People talk about high throughput proteomics, they usually talk about the speed and the scale, but that's not the hardest part. The hardest part is making sure the data stays consistent, comparable, and biologically meaningful as studies get bigger, and that's exactly what the Proteonano HT assay is designed to do. Proteonano HT Assay is our end-to-end platform for high-throughput plasma proteomics. What you are seeing here is the full process from sample preparation to protein enrichment to automated 96-well plate processing and then into the LC-MS measurement. So this is not just one reagent and it's not just one instrument step. It's a full workflow that brings together sample preparation, automation, mass spec, building quality control and normalization in one system. And that matters because in plasma proteomics, scaling up is actually not the hard part. The hardest part again is keeping the data consistent as you scale up. Once you move into larger studies, technical variations starts to come into from many different places, from sample preparation, pipetting, plate handling, LCM performance, even instrument drift over time. And if you don't control data parameters well, the technical noise can start to cover up the real biological differences you actually care about. So that's why we built a Proteonano HT Assay as a complete system. So really, Proteonano HT Assay is our way of making high throughput plasma proteomics scalable without giving up data quality. A big part of it is right here in the plate design. So it is not just the 96 samples run the same way. We actually did controls directly into every plate and every sample. So on each plate, we have 99 study samples, 3 port QC samples, 2 internal calibrators, and 3 blacks. Then inside every study sample, we also add a helper, which is short for heavy labeled peptide standards. As a quality control. So each one is there for a reason. The internal calibrator here tells us how stable the instrument is over the runs and the batches. The QCs here tell us if there's any plate-to-plate shift. The help us track sample level variation. And the blanks help us check out for contaminations or carryover over different runs. So we're not just generating data and hoping it lines up later. We are measuring variations and it happens over the whole process. And once we can see where the variation comes from, we can correct it in the right order, starting from the instrument first and then the plate and then the sample itself. So that's really the logic behind PACS. So once the plate is designed with the right controls, the next step is PACS or hierarchical normalization workflow. The idea is simple. We don't try to fix everything with one correction at the very end. So we correct the technical variation step by step in the order it shows up. So step 1 is the detection normalization. This is where we correct run-to-run variation coming from the LC-MS system itself, things like instrument drift, injection variation or signal instability over a long sequence. So we do that using the internal calibrators, if you remember that. Step 2 is the plate calibration and normalization. So here we correct the plate level variation, for example, differences caused by pipetting, reagent prep, incubation timing, or handling across different plates. This step is guided by the poured QC samples on each plate. And then step 3 is the sample level normalization. At this point, we correct the remaining sample specific variations, things like enrichment efficiency, digestion, yield, recovery, injection amount, or matrix effects. And that's where the help inside each sample becomes really important. So the final one, or at least step 4, is the inter-study adaptive normalization. This is optional, but it becomes very useful when you want to compare data across different studies, different cohorts, or want to combine data collected over longer periods of time. So we use historical QC reference to decide whether the alignment is needed instead of forcing correction every time. And then the step 5 is contamination filtering. So the picture is, or the big picture is, the packs moves from instrument to plate to sample and then across studies if needed. So that sequence really matters because each step is targeting a different source of technical variation, and that's why this work can become more than one global normalization step. We're not just making the data look more uniform, we are trying to remove technical noise in a structured way while keeping the real biological difference in the data. Yeah, so that's really the point. So with PACS, we are not just making the workflow bigger, we are making it more controlled, more reproducible, and more useful for real biological discovery.